So if you don't mind, if I could have 1 extra minute maybe, just to say thanks to Dan for inviting me. And I was really honored to be invited by Dan in particular. I was a medical student in Stanford in, I think it was 1982, and I started med school and I wanted to be a psychiatrist. I still am interested in psychiatry. Actually, on those diseases where Stanley listed, it's not, you may not realize, but psychiatric illness is incredibly common in the developing world, as it is here. But in any case, I wanted to be a psychiatrist, and Dan had mentioned how Stanley's offspring total more than 100 postdocs and grad students who he's influenced. But he also influenced a few generations of Stanford med students, and before that, med students in Seattle from his microbiology talks. And here is Stan in their first year at Stanford when they came down from Seattle. There, I'm sure you'll recognize Dan. And Stan gives an incredible microbiology course, and I knew after that course that I wanted to go and be an infectious disease doctor. And in the course of my training at Stanford, I got to know Dan fairly well on the balconies outside of Stan's lab. We would sometimes partake of some of the great herbs of California. And during our discussions then, that's one of Dan's great friends, Mike Kumi, who's a Neisseria specialist. I knew that I wanted to study also bacterial pathogenesis because Dan is an inspirational scientist, as is Stan. So that's what I have done. So the topic of my talk today, as Dan mentioned, will be cholera in Haiti. But before I launch in, I want to give you some background about cholera and the history of cholera. As I'm sure you know, depicted here is the cause of cholera. These are these curved gram-negative rods, supposedly discovered in 1883 by Koch, who's also credited with discovering the TB bacillus. So cholera, as you all do know, is a severe diarrheal disease. Infection occurs after ingesting some contaminated water, usually, or food. I think cholera is maybe the most dramatic, or at least among the most dramatic of infectious diseases. Entirely well person can get sick and die from diarrhea within a day, and develop severe diarrhea within hours. Besides diarrhea, and this is what the diarrhea looks like, vomiting is not widely known, but vomiting is also fairly common, and this makes just oral treatment sometimes difficult. The diarrheal fluid is fairly clear except for this stuff, and that's mucus. It's also often described as a rice water stool. And as I mentioned, severe dehydration can occur very rapidly. Severe cholera case can lose 20 liters of diarrheal fluid in a day. That fluid contains up to even 1 billion organisms per mil. So you can imagine, do the arithmetic, 20 liters times 10 to the 9th per mil. A single patient can really contaminate the environment and lead to the dissemination of this organism. In the 60s and 70s, I guess, there was a lot of pioneering work done, mostly in Bangladesh, where it was established that usually oral or sometimes intravenous rehydration is life-saving for cholera, and the treatment's very straightforward. Patients are put on this cholera cot, and then the volume of diarrhea can be measured, and that's replaced either by oral rehydration fluids or intravenous rehydration fluid. And with adequate therapy, the mortality from cholera is less than 1%. With poor knowledge of cholera therapy, mortality often is in the 10 to 50%. This is a photograph of a Pakistani gentleman and the 96 liters of IV fluid that were required to provide his rehydration. So cholera, as is tuberculosis, and is a disease that's been recognized since antiquity. In Sanskrit texts that date back maybe 2,500 years, there are perfect clinical descriptions of cholera. Cholera has been around outside of the Indian subcontinent for the past maybe 200 or 300 years. And in the history of cholera, which I'm going to talk about for the next few slides, 7 pandemics have been recorded. So what does pandemic mean? Pandemic means when a particular strain spreads from one continent to another. And so we're now in the 7th pandemic of cholera, which began in 1961. And I believe V. cholera is the only diarrheal pathogen to cause global pandemics. So first, before a little bit more on the history, let's talk about the history of the discovery that cholera was a waterborne disease. Stan, in his beautiful lecture this morning, mentioned some of the great figures like Pasteur in infectious disease and microbiology research. One he didn't mention was this guy here. And this guy, named John Snow, was the son of a laborer in England. And he was a truly incredible physician scientist. I'll tell you a little bit about him. So it was unusual, first of all, for a laborer to go to medical school, and he did that. And then after med school, he read a paper that was published, I think, in 1846. This is a little bit off topic, that came from Mass General, which said that you can give patients ether and do surgery. And that was just like a case report. Snow took that, having really like no background in physics or chemistry, and figured out that you could regulate the amount of gas with temperature and he did this on frogs and all these other experimental animals, and then in patients, and really kind of founded the scientific practice of anesthesia. And that led to his giving Queen Victoria in, I think, 1850 or something like that, first time a royal, this was her 9th kid, anesthesia for childbirth. So he founded the field of, really a field founder in anesthesia, but I think he's most famous because he's pretty much the father of modern epidemiology and associating patterns of disease with particular facts about human society, in this case, the use of water. So Snow first got involved in this in the 1840s or so in London. This part of my talk is germane to talk about how cholera got to Haiti, because Haiti was never had cholera, as I'll tell you about shortly, even during the 1990s when there was a profound outbreak of cholera in South America. In fact, cholera may never have been in Haiti. There were massive outbreaks of cholera in the Caribbean in the 1800s, but according to a recent paper by some Duke undergrads, there's no record of cholera in Haiti ever. So there was no cholera in London in recorded history until 1830. Then there were large, devastating outbreaks of cholera in England and throughout Europe. In fact, cholera is the premier infectious disease of the 19th century. Plague was the 16th century, 19th century was cholera. And we have to put this in context. How did they think about infectious disease in 1830s and 1840s? The ruling hypothesis was not the germ theory, it was the miasma theory. I don't even totally understand what that means, but what that means is something about bad air gave rise to infectious disease, and that was always inextricably linked to some sort of theory of the people who the bad air was around. Usually that's poor people, in the case of the way people thought about disease then. So that's what was the intellectual milieu where Snow was brought up in at the time. But he noticed, so there was devastating cholera in the 1830s. Then there was a period of several years where there was no cholera in England. Then in late 1840s, I can't remember the year exactly, 1848 or something, there was a big outbreak of cholera in Hamburg, Germany. And then there was several months later an outbreak of cholera in England, in London. And in fact, what is most likely the index case came from Hamburg, went to a hotel in London, and he died of cholera there, and that was like the first case in several years. The next occupant of that hotel room also died of cholera. And then in the subsequent weeks, that's how led to this 50,000 deaths over the next 2 years in London alone. And that led Snow to first speculate that this disease is caused, first of all, by human transmission. It was very hard to imagine how the miasma might have traveled from Germany to England. That's just unfathomable. His ideas then were not accepted, but let me just read to you from this paper that he wrote at that time. Which is this particular book is a terrific book, which I recommend to you, called The Ghost Map, about Snow in London in 1854. Who can doubt that the case of this guy who died, John Harnow, the seaman from Hamburg, was the true cause of the malady in London? He came and lodged and slept in the only room in all of London in which there had been a case of true Asiatic cholera for a number of years. And if cholera be communicated in some instances, is there not the strongest probability that it is so in others? In short, that similar effects depend on similar causes. People didn't accept that paper. And there were some reasons that you could argue, but his next work was more accepted. And this was in the outbreak that occurred in 1854, several years again later. And during that outbreak, what Snow did, and this I'm sure many of you are familiar with, this was published in 1855, make a map of the distribution of the cases of cholera in this neighborhood in London. And what he found here's where people got their water from this pump on Broad Street in London, that many, many, many cases were around here and not in the surrounding neighborhoods where people got water from other sources. And furthermore, there were cases in some neighborhoods that were fairly far away, but he tracked those cases, and those people got their water from the Broad Street pump, which was a very compelling argument. And then some people who didn't get sick right around here, the epicenter, ultimately didn't get their water from there. So it was a pretty persuasive argument, didn't kill the miasma theory, but I must say, overall, Snow's arguments, we didn't think in retrospect even about John Snow's discoveries for cholera traveling from Hamburg to London when we were thinking about the origin of cholera in Haiti. So as you know from the story, Snow convinced the local magistrates to close the pump, and that stopped the epidemic. And this is a famous lithograph of here's the pump and death's dispensary. Okay, so let's talk about the organism now. It's a gram-negative highly modal rod, there are more than 200 serogroups of cholera, and these are based on the chemical composition of the outer membrane lipopolysaccharide. In only the O1 serogroup has caused pandemics, it's thought. However, the O139 serogroup has caused epidemic cholera in South Asia since 1992. There are two biotypes. This is based on a variety of phenotypic classifications that distinguish the O1 serogroup, classical and L-Tor. And so here we'll go through the world history of cholera on this Google map. So the first 6 pandemics are thought to be caused by this classical biotype. They all arose in South Asia and then spread to Africa. This map, I'm not exactly sure where I got it, is not correct. Cholera also came to South America, the Caribbean, Latin America, and the US. During the 19th century, there were profound outbreaks of cholera, particularly in New York City. Then, in 1961, there was the beginning of the current ongoing 7th pandemic of cholera, began in this Indonesian island of Sulawesi, rapidly spread to the traditional home of cholera in South Asia, and then in the 70s spread to Africa, where currently most cases of cholera reported to the WHO come from Africa. This 7th pandemic clone, L-Tor clone, arrived in South America in 1991 and then spread throughout Latin America, including into Mexico, in the following decade, where it caused more than 1 million cases and more than 10,000 deaths. However, in the past decade or so, it's pretty much burned out. But for this talk, there was no cholera in the Caribbean from this, including on the island of Hispaniola, which you know is where Haiti is. So I'm going to end on this historical note here and say that this book, which I mentioned to you about snow in London, is terrific. An equally, maybe more terrific book, which actually Stanley recommended to me like 15 years ago. I never read it then, but now have looked at it, is this book called The Cholera Years, about cholera in the US during these 3 outbreak periods. And this book is written by Charles Rosenberg, who, for the gossip point of view, is married to the president of Harvard University, Drew Faust. The main point of this book, which I think is kind of a field-founding book, is how our thoughts about infectious diseases, in this case really diseases overall, are so determined by the intellectual milieu of the time. So in 1832 in the US, when cholera first came, the outbreak was thought of as something that happened to sinful humans, usually sinful poor humans. And by 1866, it was recognized that this was related to, partly from the work of Snow, deficiencies in our sanitation system. I think there are some key lessons here from all this thinking is that, and I don't want to insult anyone here, but the Republican idea to dismantle the government and various civil activities goes against a lot of human history about at least the value of keeping our shit away from our drinking source. I'll briefly talk about the virulence or pathogenesis of this organism. This organism is not an obligate pathogen. It's happy to live in aquatic reservoirs with a variety of different marine creatures. We get cholera from ingesting contaminated food or water. Then it has the unusual capability to live in the small bowel and where it elaborates a toxin. That toxin, cholera toxin, is sufficient to cause cholera. There's been human studies, actually in the United States, where given purified cholera toxin to volunteers in Maryland, Mike Levine showed they get cholera. So this is a purely toxin-mediated disease. So the main virulence factors of this organism were both discovered in the laboratory of Mecolanos, or not cholera toxin, but the genes for cholera toxin. But this pilus here enables the organism to grow in the small bowel, and this toxin here, there's the cholera toxin crystal structure. They're both interestingly carried on mobile genetic elements. Stanley alluded to in his talk that many, perhaps really most, maybe the vast majority of genes implicated in virulence are oftentimes not part of the intrinsic backbone of pathogens, but rather part of a mobilome, if you will. Interestingly, the genome of Vibrio cholerae, which was first reported in 2000, is divided between 2 chromosomes. All the Vibrionaceae have 2 chromosomes. And as I mentioned, so the TCP pilus is encoded right here in a pathogenicity island. Cholera toxin is encoded here in a filamentous phage, which I'll mention to you in a minute, Dan alluded to. And also another, not virulence, but antibiotic resistance cluster that accounts for most antibiotic resistance in clinical Vibrio cholerae isolates from around the globe is encoded right here in an integrative conjugative element. It's kind of like a plasmid, transfers by conjugation, but integrates into the chromosome right there. Just to toot my own horn, I discovered both of these mobile elements when I was a postdoc in Mecolanos' lab, and analysis of these 2 components of the genome helped us decipher the origins of the Haitian strain, which I'll get to in a minute. So, as Dan mentioned, cholera toxin is encoded by this weird-looking phage here. Most phages that you think of are kind of shaped like lunar landers, like lambda, for instance. But cholera toxin phage is just a polymer, a protein polymer with single-stranded DNA inside, and at either tip there's a few little proteins. We just, for the first decade or so that I had a lab, we worked on many of the molecular mechanisms, how this phage works. Interestingly, its receptor is the TCP pilus, which is that colonization factor that enables Vibrio cholerae to colonize, suggesting the possibility that TCP, which is expressed in the gut may have been the site where toxigenic Vibrio cholerae evolved. It has many very cool mechanisms that it mediates its life cycle, but they're not really germane now. Let me say a little bit, though, about this SXT element. We first discovered this in the first non-01 strain, Vibrio cholera 0139, in a strain from Madras, India. We now have recognized that this element transfers by conjugation. However, it does not replicate autonomously. It replicates with the chromosome after it integrates site-specifically. Now, wherever we isolate Vibrio cholera and other related Vibrios, we see this SXT element, and we've sequenced, first initially, a grad student sequenced this 100 kb element. This guy actually went on to do a postdoc with Dan. Now we've sequenced these elements from around the world from Vibrio isolates, and they are mosaics. And for this talk, what's important is that we can see the constitution. We can use the content of these antibiotic transfer elements as a relatively specific signature for where they come from on the planet. Okay, that was a fairly long introduction, and now we turn to Haiti and a few words about the pre-epidemic period there. I had mentioned to you already that the 7th pandemic never made it to Haiti. As I'm sure you all know, Haiti is the poorest country in the Western Hemisphere. The WHO evaluated the water supply in Haiti and said access to water, safe water, remains uneven. According to WHO, 12%, this is in 2008, 12% of Haitians received piped water at that time. And yesterday is the 2-year anniversary of the earthquake in January 2010. This killed now, I think what they were saying yesterday was 315,000 people and left 1.5 to 2 million people homeless. Amazing damage to the already lousy infrastructure. One thing to note, here was the epicenter of the earthquake. The cholera epidemic did not begin in this area. As I'm going to show you shortly, it really happened along a river right here, right around here. Now, I would like to read to you this a little bit of a long quote from the CDC, who in March of 2010, the CDC analyzed what the risk of cholera might be to Haiti after the earthquake. And that what they wrote was the following: an outbreak of cholera is very unlikely at this time. While the current water sanitation infrastructure would certainly facilitate transmission, cholera is not circulating in Haiti, true, and the risk of cholera introduction to Haiti is low. Most current travelers to Haiti are relief workers from countries without endemic cholera. Simply not true, and they are likely to have access to adequate sanitation. So that's our US CDC, and this statement may in fact hold them somewhat culpable, this analysis, because in fact, most security forces in Haiti and around the planet come from cholera endemic regions. So this is how wrong they were. This is the epidemic curve through October. Here's the original peak in October 2010, and now this is up to October 11. And then I just updated this from the Haitian Ministry of Health through Christmas of 2011. There's been more than 500,000 cases and more than 7,000 deaths. So the epidemic is thought to have begun on around October 19th. Now the index case is known, we may get to it, happened about a week earlier. And this was made public about October 22nd, that there was a cholera outbreak in Haiti. And the CDC got these strains about a week later. I contacted them because my lab had a fair amount of experience using whole genome sequencing to map suppressor mutations in Vibrio cholerae, and I offered them our expertise to analyze the genome of this strain, and they were first very game to collaborate with us. But in the next week or so, this spread, the outbreak spread, and it also became politically very hot, and they no longer return my phone calls. In part, it became hot because Haitian people thought that the UN peacekeepers, this is the Haitian people themselves, may have brought cholera there. In the outline, by the first month, all 10 states or departments of Haiti had cholera. And these were fairly large demonstrations, and people got shot in these demonstrations, actually. It became very tense. Right when this broke out, many cholera researchers from around the globe, my lab, other labs, began to wonder, well, how did cholera get to Haiti, which is the question for most of the remainder of my talk. And several hypotheses were put forward. The one that I favored was some kind of environmental conditions enabled Vibrio cholerae to get from the Gulf of Mexico, let's say Mexico, over to Haiti. They already had cholera there. That seemed most likely. Another possibility was put forth by a fairly well-known cholera researcher, Rita Colwell from University of Maryland, and she proposed that native non-pathogenic and endemic strains evolved into pathogenic strains, that cholera was basically autochthonous to Haiti, and this was de novo evolution. To me, that sounded kind of crazy, but with the existence of CTX phage, you could imagine that a toxigenic strain might arise anew in Haiti. And another possibility, which I must say, in retrospect now, that we were stupid not to even consider, was that cholera was brought to Haiti from somewhere else on the planet by a human, just like John Snow figured out in 1848 in the Hamburg to London transmission. So after the CDC said they would no longer share strains with us, I was lucky to contact colleagues from MGH who actually had 2 physicians in Haiti helping to treat the work in the epidemic, and permits and everything were acquired, and we got these strains at Harvard on November 8th. At this time, once I knew we were getting the strains, I started a collaboration with the California biotech company, Pacific Biosciences. I didn't really know how good this would turn out to be, but this was incredible luck. I'll go through what their technology is in a second. But basically, during a graduate committee meeting, I got a phone call from their CSO. This was a Monday morning, and he interrupted that meeting and said, okay, his name is Eric Schott. He said, we're going all in. So when we got these strains, someone in my lab, Yoshi Yamaichi, we got these on the 8th. He stayed late that night. And PacBio going all in means they sent a courier at 4 a.m. to Longwood Avenue and at Harvard Med, and they picked them up, and they took them to Mountain View, a little bit down the road. And then later that night, data was streaming off their machines. And in retrospect, they say that they could get the answer that this really came from South Asia, as I'm stealing my own thunder, in about 2 hours. I don't know if they're right about that, but it was certainly within a day or two. So this was a very collaborative project. That included people from PacBio, led by Eric Schott, people from my lab, Mekolanos' lab, and the doctors from MGH. Here's what our study design was. We characterized 2 Haitian genomes, a genome from Peru from the outbreak that began in '91, because we thought maybe the Haitian outbreak actually came from the Latin American outbreak. And then 2 Bangladeshi isolates. One was from 2009, a recent one, and one from 1973, which was a resequencing of the canonical strain that we all use as a reference strain. And what we did was to analyze both SNPs around the chromosome as well as the structure of hypervariable elements like the SXT element. And we used it for this project, PacBio, they call it single molecule real-time sequencing technology. I believe it is. I have no interest in PacBio stock or anything, but it is very exciting. I'm going to just talk about that for a second. Current next generation sequencing technology, the ruling platform is Illumina. I'm not trying to put it down. Illumina is incredible. We do a lot of sequencing with Illumina. We are now routinely getting 350 million reads per lane. That's just too hard, too great to imagine how for bacteria it's not necessary. The main principle in Illumina, it's called a wash and scan kind of technique. So it's DNA sequencing by DNA synthesis. This takes place on a plate where DNA is synthesized, and then after each nucleotide is incorporated, the dye gets cleaved, washed, and then it happens over and over again. As you all know who have done Illumina sequencing, it takes at least a week to get the data back. It's super deep, but it takes a little bit of time. PacBio is also sequencing by synthesis. However, it works in real time. So what happens is, I don't fully understand it, but there are the nanowells etched into a plate, and single DNA molecules and single molecules of DNA polymerase from a phage they get to go into individual wells, and then DNA is incorporated. And as it's incorporated, each incorporation is read. and that's so you get the reads in real time. So that's a cool thing. Another cool thing about PacBio is that you not only get the sequence ATGC, you also get the time it takes to get incorporation. And I'm not going to have time to talk about this today. This is ongoing work. But in the next 6 to 8 months, I think we are going to have some truly revolutionary data because we can now do genome-wide measuring of the epigenome in bacteria and mitochondria based on using the kinetic variations that we see in this kind of work. Finally, another excellent feature of PacBio is the read length. Currently, the read length for Illumina is something like 100-ish. PacBio now, in this study, was 900. But now we also did a similar project with E. coli 104. It was 2 kb. Now we're up to 5 kb reads, which is super cool. Great for getting resolving repeat regions in the genome. OK, a downside of PacBio is it's not as accurate as Illumina. So we got these reads back in basically kind of 6 hours per genome. We had 2 Haiti strains, H1 and H2, a contemporary Bangladesh strain, the old Bangladesh strain, and the Peruvian strain. So one great challenge for all biology now, especially high throughput biology, is how to actually visualize data when you have, for each one of these genomes, 4 million base pairs, and then you have 5 genomes here or 10 genomes. You have hundreds, in the end, hundreds of millions of base pairs. How do you look at the data? Snow was a genius because he introduced a new way to look at data, superimposing on a street map where cases were. And that led to his figuring out this is waterborne. And we still are challenged now in our epoch of high throughput biology to really see data. So I recommend to you, undergrads, grad students, even postdocs, to think about learning at least some computer stuff to help you out to look at your data in the future. So how did we look at this data? First, what this represents is called a coverage map. So for each one of these genomes here, we had something like 20 to 50x coverage. And then we plotted the coverage against the reference genome, which was the same as this strain here. And so this was basically identical to the reference genome. And then wherever there was a gap, you see these little red lines. That means those bases weren't there. And so you can see here, for instance, this is a pathogenicity island, that the 2 Haiti strains that we sequenced, which were essentially identical, were missing this or had a large deletion in this pathogenicity island that was present in the canonical reference strain. It was also seen in the recent Bangladesh as well as in the Peruvian strain. The SXT element I'll get to in a minute. And then here on the second chromosome, there was again, this should be orange, some orange bars indicating a deletion in the superintegron, which is a large gene capture system on chromosome 2. So you can see that these have identical deletions, which is not present in the Peruvian strain or in the canonical reference strain. I think one of the key elements of the analysis then of where these strains come from and what they're most related to came from looking at all the conserved genes in the pan-Vibrio cholera genome. And there was about 1.8 megabases, or 1,600 genes, that we saw in all sequenced V. cholerae. This included L-Tor strains, which are shown up here, as well as other Vibrio cholerae, including classical. And then we took all the SNPs in these 1,588 genes and made a phylogenetic tree. And what you can see here are several key things. One, the 2 Haitian strains are identical, and that they're part of this whole group of L-Tor 01 Vibrio cholerae. They're part of the 7th pandemic. But their closest relatives here out on this branch are two recent Bangladeshi strains. This one here we sequenced. And they're quite far away from the old canonical reference strain as well as from the Peruvian strain in 1991. So cool. This provides strong evidence that the Haiti strain is highly related to the South Asian strains, recent South Asian strains. Then I mentioned to you the superintegron on chromosome 2. Now this is just an alignment of the ORFs. Here's the canonical reference strain, the ORFs. The Haitian strain had an identical 41 kilobase deletion as seen in this recent Bangladeshi strain, whereas the Peruvian strain had all the same ORFs as the canonical strain, so quite different. Similarly, the SXT element in the Haitian strain was again identical to these recent Bangladeshi strains, and in fact, the SXT element was not in the Peruvian outbreak strain, which had never been sequenced, nor was it in the canonical reference strain. So for time, I think I'm going to skip over that and then get to a couple of conclusions from this part of the work. So I had 5 minutes. Is that what that meant, right? So first, I think these data strongly suggest that the outbreak bears striking similarity to El Toro 1 strains from South Asia. These are called variant V. cholerae El Toro 1 strains. And they're very distinct from strains from Latin America. And this suggests the most likely explanation for the appearance of cholera in Haiti is transmission of V. cholerae by an infected human, most likely, but it could be some other contaminated item, and argues strongly against our first hypothesis, which is that it came from Latin America, and I think argues against the Colwell hypothesis that this was an autochthonous de novo generation of a new strain. What about the idea that cholera actually came from the Nepalese troops? We tried to get 2010 Nepal isolates, but we were unable to. But subsequently, people have gotten them, and I'm going to show you their data in a minute. But this guy, Pirot, a French epidemiologist, did a traditional John Snow-like shoe leather epidemiology after the outbreak, and he put together this graph, which I'll show you, of the timetable as well as the geography of the beginnings of the outbreak. Right here is where the outbreak began along this river, and the very first cases of cholera, there was a paper published online yesterday that the index case was October 12th, which took place. Here's where the UN Nepalese troops were stationed, right here. This is in this blow up here. Took place, this first case right here. That's where the index case probably occurred. The timeline and where this happened is very consistent with the idea. And ultimately, the UN evaluate themselves whether cholera came from Kathmandu, and they have embraced that conclusion. There was excellent journalism that showed that the UN waste material was dumped into this river, rivulet, whatever, right there. And now there's sequence data that strongly suggests that there is essential identity between these Nepalese 2010 strains, this particular branch here, Nepal 4, and the Haiti strains. I should say, I didn't point out here, that there was cholera in September 2010 in Kathmandu, and on October, that's when the first Nepalese soldiers came to this area where the first cases occurred. So it's pretty good data. I think we can then conclude with some other conclusions from this work, I think. In the future, travelers, relief workers, and security forces from areas where cholera is endemic should be screened and/or treated with antibiotics, with vaccines to prevent introduction of cholera to regions which are high risk. Remember, the CDC thought that relief workers and security forces don't come from endemic cholera areas. That's just simply not true. We had written this in our New England Journal paper, and just to show you how this idea, how conservative the editors of the New England Journal are, they made us take this out because they thought that was an inflammatory conclusion. Another, I think, lesson from this outbreak is that stockpiles of cholera vaccines, which are not terrific, but at least pretty good, should be available. Very early on in this outbreak, I and other people were very strong advocates for using vaccines. And it turned out I talked to the companies. There's one company that made a cholera vaccine. On the planet, this planet, the time of the outbreak of cholera in Haiti, there were less than 200,000 doses of cholera vaccine available. So it wasn't even that reasonable to think about applying vaccines. I think another conclusion is that we're on the cusp of a new era of whole genome-based molecular epidemiology and sequencing technology. Just 2 days ago, there's a report in Nature Nanotechnology about an entirely new genome sequencing technology based on nanopores and nanowires. I don't know exactly what that means, but the bottom line conclusion of that is that Valerie now in 6 years is going to go to a TB patient, put her iPhone on there, and then get the sequence. That's not a joke. And I did have a whole nother, like 5 slides to talk about cholera vaccines and our work we've done to make an improved cholera vaccine, but I blew my time a little bit on waxing about history, but that's kind of fun anyway. I do want to get to the acknowledgements. The epidemiology work was a terrific collaboration between my lab, Mekelanos' lab, doctors from MGH, and especially from Eric Schott's team. He's now the head of genomics at Mount Sinai University. There's my lab. I know it may not be a major football school, Berkeley, but there's the quarterback of the Pats. Dan said that that was true. I really don't know. I'm rooting strongly for the Pats. I do hope that this Tom Brady, who's a graduate of the University of Michigan, will face that eminent Berkeley graduate, Aaron Rodgers, quarterback of the Packers in the Super Bowl. Thank you.
Questions? Right there. Real quick math, though. Does this imply that there was a carrier state in the workers?
So the UN and the Nepalese troops have vehemently denied that anyone had cholera. So if you take that at face value, then I think there is some carrier state. It's not like salmonella, chronic carriage, but definitely people who have had cholera or even asymptomatic infections, you can culture cholera from their stool for up to 3 or 4 weeks after initial infection, which is totally consistent with the timeline.
So I have 2 questions. Given that this is a global health symposium, I'm wondering if you have some thoughts regarding the fact that Haiti is 2 hours from Miami, that cholera is completely treatable with only IV fluids, and how we ended up with 7,000, and that Haiti had the largest infusion per capita of AIDS probably in the history of the world in the year after the earthquake. How did we end up with 7,000 deaths from cholera in Haiti? So that's the conjectural.
Okay, so stop there. This symposium is actually called something about diseases of poverty, and I think that's the real key thing. is that Haiti is unfathomably poor, and delivery of health care, most cholera deaths have happened in rural areas where people can't even get access to health care. So in the beginning, the death rate was over between 5 and 10% in the first few weeks. Now it is down in the less than 1%. So people are getting treatment, but it was a terrain that was more than perfectly ripe to have a outbreak such as this. But you're right, I mean,
Okay.
it seems
So the
unfathomable.
second is the completely other extreme, and this is actually for you and Stan. If you look at travel medicine, rarely do you ever see a traveler get cholera, and it's presumed that that's because it takes about 10, the ID50 is about 10 to the 9th organisms, whereas at the other extreme you have Shigella where it's 10 to the 2nd, and Salmonella typhi, say, 10 to the 5th to 10 to the 7th. Do we have any understanding of why we have such widely variant infectivity rates per organism with these 3 different enteric diarrhea-causing bacteria?
Short answer, no. But Steve's asking a very interesting question. For a patient to get Shigella, maybe one organism may be required. For cholera, for white boys from Maryland, yes, it takes 10 to the 8th, 10 to the 9th. That's probably not the case in a malnourished, achlorhydric people, like most people in the world in Bangladesh, where it's maybe 10 to the 5th, maybe even lower. But still, we don't really know what are the limitations of infectivity.
I'd like to make one more comment about the Haitian outbreak. A lot of the first patients were seen at the Albert Schweitzer Hospital, which is supported partly by Stanford. And there is no clinical microbiology laboratory, even today in all of Haiti, that does a standard stool culture and would have recognized that organism.
That was great. Yeah, thank you, Matt. I was the concept that cholera is kind of accidental in humans is I used to think that, but now I wonder if you think that maybe cholera actually uses humans as part of its life cycle for amplification, since I don't know what its real host may be in the real world. With all the stuff on crustaceans and diatoms, but it seems to me that it gets its major amplification, certainly in the Bengali area where it began, by going through humans.
I think from the historical point of view, at least that's what this book strongly argues, that the history of humans getting together in cities is instrumental for allowing Vibrio cholerae to become such a profound pathogen. I don't know if it's a requisite part of its life. I think that remains to be seen.
Does cholera toxin have any other, in any of these other models, does it provide any kind of advantage to the bug?
Oh, that's not showing anymore? Oh, I got the coolest movie here. I was showing you the bugs with 2-photon microscopy.
So while you're doing that, I wanted to mention about the infectious dose. I would say one thing. Infectious doses of pathogens are done, as you say, on white boys in Maryland who volunteer, usually medical students who need the money. But also, it depends on how you grow the organism. And we grow the organisms in the laboratory at 37 degrees and funny complex media. And I'm not, if you grow salmonella correctly, the infectious dose drops from something like 10,000 or so down to closer to 1,000. And when you realize in natural infections, say waterborne typhoid, when you go and look for the number of organisms, it's bloody small. So I think a lot of our views of that are still are tainted, as it were, by not doing the right experiments. Are you ready now?
I know my time's up, but this is a 2-photon microscopy, basically intravital microscopy of Vibrio cholerae, making a microcolony down here on the intestinal villus. Just had to show that. And it's super fast organism. Anyway. Yes, that's real time.
One last question.
What is the effectiveness of the existing cholera vaccine, and what's the effectiveness of your new vaccine?
And what is the basis of the vaccine?
Okay. So the current licensed vaccine, there's 2 forms, they're basically the same thing, are killed whole cell vaccines. Grow up the bug, kill them, phenol or heat. One of them has B subunit of cholera toxin, the other one doesn't. Those need to be given in 2 separate doses, spaced out by 2 weeks. The effectiveness at 1 year is something about 60%. There are live vaccines, first pioneered by Mecolanos and by Caper. The Mecolanos vaccine in human volunteers has been put into a couple thousand people. It's safe. Some people get mild diarrhea. That's what I was going to show you, how we can eliminate that reactogenic diarrhea by deleting the flagellins, which activate the innate immune system. We don't know the efficacy of those vaccines in the field. The Caper vaccine, CVD103-HGR, was tested in Indonesia. It had no efficacy, 0. They had very little cholera. So we need to do more clinical trials. I think really we can do so much better than the killed whole cell vaccine. And that's one challenge I will put to all the students and postdocs here is make new vaccines. They, as Stan showed in his work, they really work.
All right. Well, I'm glad you didn't go into psychiatry, although I think you need one. Okay.
We all do.